Ladies and gentlemen, thank you for standing by. Welcome to the Pluristem Analyst and Investor Call. All participants are present in listen-only mode. A question and answer session will follow the formal presentation and will be accessible for analysts only by clicking the raise hand option on the bottom of the screen. Other participants, please submit your questions by clicking the chat box. With us on the call are Mr. Yaki Yanai, Pluristem CEO and President, Dr. Nitzan Halevi, our Chief Medical Officer, Chen Franco Yehuda, CFO, and Dr. Jacob Rowe, member of our advisory board for the HCT trial for the Hematology at Shari Tzedek Medical Center, Jerusalem. Before I turn the call over to Mr. Yaki Yanai, the company's CEO and President, I will now take a brief moment to read the Safe Harbor Statement. During the course of this call, management will make express or implied forward-looking statements within the Private Securities Litigation Reform Act of 1995 and other U.S. federal securities laws. For example, we are using forward-looking statements when we discuss the PLX cells may provide a safer and more effective therapeutic benefit than current technologies that PLX-R18 may potentially enhance. The production of 3 blood cell lineages that PLX-R18 may potentially reduce transfusion dependency, the belief that PLX-R18 affects the regeneration activity of the hematopoietic cells, our intention to push forward with the clinical development of PLX-R18 with the goal of establishing it as the new standard of care in the field of and the expected timing of the expected end of enrollment in our various studies. These forward-looking statements and their implications are based on the current expectations of our management only and are subject to a number of factors and uncertainties that could cause actual results to differ materially from those described in the forward-looking statements. Except as otherwise required by law, we undertake no obligation to publicly release any revisions to these forward-looking statements to reflect events or circumstances after the date hereof or to reflect the occurrence of unanticipated events. For a more detailed description of the risks and uncertainties affecting us, reference is made to our reports filed from time to time with the Securities and Exchange Commission. I would now like to hand over the call to Mr. Yaki Yanai. Mr. Yanai, would you like to begin?
Thank you very much. Good morning, everyone, and thank you for attending this call today. I'm very glad to present today the top-line results from our hematology study. This is a phase 1 open-label first-in-human dose escalation study. The main goal of the study is obviously efficacy for IM injection, safety for IM injection of PLX-R18 with patients with incomplete hematopoietic recovery following bone marrow transplantation. We do want in this presentation today to give you insights also to the 6 months follow-up exploratory efficacy endpoints that we're going to present and discuss. So let's start to discuss the patient population. So who are the patients that we were targeting in this indication? Our patients are patients after bone marrow transplant. While they had some engraftment, they are still suffering from a poor function of the graft. These patients, because they poor function, they are very high-risk patients because the body fails to develop a sufficient blood counts. They are suffering from very low blood counts or bloodlines. The patients, all of them are treated with the current standard of care. That most of the standard of care that we are familiar with is targeting most of the time one of the bloodlines, and by that it makes it quite limited. Our model and concept is to targeting the regeneration of the hematopoietic niche in the 3 bloodlines. Our patients are very vulnerable patients, and most of them will be on continuous infusion of blood products on a regular basis during the current treatments, during as part of the treatment of the disease. PLX-R18 is one of 2 placenta-derived products that Pluristem is developing. But the PLX-R18 is a product that we have developed for specifically targeting hematological deficiencies. The ability of PLX-R18 placenta cells to secrete a variety of a cocktail of therapeutic proteins that are very relevant for hematological recovery makes it a very good agent for this situation. This is a safety study, but as I mentioned, we do want to see the exploratory efficacy endpoints in our study. We have developed this protocol and designed this indication based on very robust preclinical studies that we have developed for years with the NIH and the DoD. This product, the PLX-R18, is also part of the ARS, Acute Radiation Syndrome project, and we did quite a lot of, and collected quite a lot of data with the NIH and the DoD, seeing the ability of the cells to recover the hematopoietic niche and to support the recovery of the free blood lineages. In this phase one study, its main purpose is to see the effect of the PLX-R18 on humans, and to see the potential of our product to stimulate and get to realize the therapeutic potential of the transplant, the bone marrow transplant, and to lead the recovery of the patient. The goal here is we wanted to mimic and to see what we had in preclinical support of the hematopoietic niche and the recovery of the free bloodlines, which is different from the current standard of care. The PLX-R18, like all Pluristem products, is manufactured in our in-house GMP manufacturing facility. We are using the three-dimensional technology, which gives us the ability to manufacture in a very robust batch-to-batch, high-scale manufacturing process. But what is unique about this product, and I'm glad to present that, this is the first product that we are presenting, which is fully serum-free media, which is developed and owned by Pluristem, and we were able to show a full comparability with previously designed PLX-R18, supporting the quality as well as the potency of the product, and that tells you the ability of Pluristem to control all the supply chain from raw material to the patient bed, which allow us to manufacture the products very efficiently. We see the PLX as a platform. I'm sure that most of you are familiar with the PLX-PAD, which we are using today in advanced clinical studies. The PLX-R18 is presenting today the first in-human clinical data. These are like two strong APIs that we have developed over the years. We do have the capacity, and we presented in the past products like PLX Immune, which are induced products. They are based on the two PLX-PAD and PLX-R18. And by seeing the PLX Immune, that actually opens the door for a future product which are tailored, designed for specific indications. Our next generation product is a product that is derived from placenta on the PLX-PAD and the PLX-R18, but it's genetically modified using the CRISPR technology. We are part of a consortium that is developing this product, and the concept is to develop a library of almost unlimited number of modified products target for specific indications. I would like to use this opportunity also to give a brief on the current pipeline of Pluristem and status. The phase 3 study for muscle regeneration is about 85% enrolled, and we are expecting to continue to complete enrollment in the coming quarter. In the ARDS studies, we're having 2 studies, multi-center, multinational studies, US, Europe, and in Israel. We expect to complete enrollment in the coming quarter for the European study and the US study during H2 2021. We are continuing the recruitment for the chronic GVHD product. This is a physician-initiated. We just recently moved to the second cohort, and we are continuing to enroll patients for this study. And with that, I would like to hand the presentation to Dr. Nitzana Levy, our Chief Medical Officer. Please, Nitzana.
Thank you, Yaki. So it's my pleasure today to walk you through the 6 months result for our phase 1 study that was conducted in patients with persistent deficiency of at least one blood cell type following bone marrow transplant. As Yaki mentioned, This is a phase 1 first-in-human study, and the key primary endpoint was safety. And I'm happy to share with you today that based on the data collected so far, the PLX R18 was well tolerated and demonstrated a favorable safety profile. As for exploratory efficacy, I'll show you in the next few slides, our 6-month data, all available data at 6 months, showed significant improvements in all 3 blood cell types: platelets, red blood cells, and white blood cells. Also showed a substantial reduction in transfusion dependency. A quick overview of the design of the study. So this was an open label dose escalating study that recruited patients with incomplete recovery at least 3 months following their bone marrow transplant. To be included in the study, the patients needed to have at least one of the 3 blood cell lines below a pre-specified threshold. This threshold was 50,000 cells for platelets, less than 8 grams of hemoglobin, or less than 1,000 neutrophils. We enrolled 21 patients in centers in the US and Israel to 3 cohorts. The first cohort, low dose, 1 million cells per kilogram, enrolled 3 patients. The second cohort, 2 million cells per kilogram, included 6 patients, and our high dose, 4 million cells per kilogram, included 12 patients. Again, the pre-specified primary endpoint was safety, but we also had pre-specified exploratory endpoints that talked about changes in all the 3 blood cell types from baseline to 6 months and to 12 months, and transfusion dependency. The duration of follow-up for each patient is 12 months, and today I'll share with you results, the complete results for the 6 months follow-up based on data from 14 patients. Of the 21 patients that were enrolled into the study, 7 don't have data at month 6. This includes 3 patients that had fatal events, 1 on the 4 million per kilogram dose and 2 on the 2 million per kilogram dose, and none of these fatal events were considered related to the study treatment. 1 patient early terminated prior to the month 6 visit, and 3 additional patients didn't have a month 6 visit, so we don't have data to present today. The baseline characteristics show a median about 8 months from their bone marrow transplant to the first treatment, 236 days, despite the fact that they were able to be recruited as quickly as 3 months after the bone marrow transplant. Actually, this value is about 8 months on median. The mean age was 56 years, and that most of our patients received an allogeneic transplant, 90%. And now to the results. What you see here are improvements on all 3 blood cell lines for the 3 doses. So to the left, you see the mean hemoglobin levels from baseline to month 6. In the middle, the neutrophil counts, and to the right, the platelet counts. As far as the color charts, the red lines indicate the 1 million dose, the 1 million cells per kilogram dose, the green, the 2 million cells per kilogram dose, and in the blue, our high dose of 4 million cells per kilogram. You see that for all 3 cell lines and all 3 doses, we saw improvements from baseline to month 6. However, I'd like to point out that for the high dose, these changes were statistically significant from baseline to month 6 with a p-value of less than 0.0. Looking at this data a little differently and trying to understand how patients responded to treatment based on whether they were below or above the threshold on 1, 2, or 3 parameters as I defined previously. So you see on the bar graph to the right, our left graph shows pre-treatment values. We had 5 patients with 1 parameter below the predefined cutoff, 11 patients that had 2 parameters below the predefined cutoff, and 5 that actually had all 3 parameters below the predefined cutoff. And at 6 months, you see that 10 of the patients had all 3 parameters above the predefined cutoff, which is a very substantial response. And last but not least, I want to share with you the data about the reduction in transfusion dependence at 6 months. Of our 21 patients, 13 were transfusion dependent at baseline. And at month 6, only 7 are considered to be transfusion dependent. Now, importantly, the analysis here also includes patients that failed for other reasons or we don't have data on, and not only those that are actually transfusion dependent. So this is a quote unquote intent to treat analysis. With this, I'd like to hand over to Dr. Jacob Rowe to provide clinical perspective.
Thank you very much, Dr. Lev. Let me say, just start off by a word by describing the kind of clinical scenario we're talking about. And this is a graft failure after transplant, but it's not what's often known as graft failure. Typical graft failure is after transplant, within a month, maybe 5 weeks, patients do not engraft. And that's a different group, not the one that we're discussing today. That's classically the word. What we are talking about is patients who initially engraft, seem to be doing well, but then they start limping along. Their blood counts go down. We check their bone marrow. There is engraftment, but then they limp along. Now, that's a difficult patient population to treat. This is a population that has frequent outpatient visits, regular hospitalizations, and multiple complications. Now, as you saw, this is a heterogeneous population, has multiple diseases. It's uncommon in any single indication. Altogether, we are talking somewhere between 5 to 8% of all transplants, although it seems to be increasing now as we're transplanting more older patients and thus more with comorbidities. So there isn't a series in any one center. You saw there were some patients with acute lymphoblastic leukemia, some acute myeloblastic. These make up the totality when we give here the numbers, but that's a clinical scenario, and that fills up a great proportion, even the small numbers, of the outpatient and inpatient visits after a transplant, certainly in terms of cost. I can tell you, as a transplanter for more than 35 years, this is what takes a major part of our clinical effort. So it's a real population. So there is no question, this is an urgent and a historically and a current unmet need. Don't know how to deal with this. Now we're talking about patients who've had all the treatments. They've had cytokines of GCSF, they've had erythropoietic stimulating factors, everything else, but they still have the problem that we have here. Now, again, this is a phase 1 study, and a phase 1 study, as we know, needs to look at the study. And when we have a product that's never beforehand been used in humans, it's very reassuring to know that we do not have a safety issue here. What about a signal of efficacy? And I'm going to be careful. I say the word a signal of efficacy because this is not a controlled study. Some patients, no doubt, would recover spontaneously. There's some recovery. I cannot tell you. But we're talking here, you need to remember, this seems to have been an entrenched problem. The median time has been at least 8 months from transplant, with a mean close to 10 months. In fact, it occurred less than 4 months from transplant. That was earlier. So we really are talking about a patient who seemed to develop, they've sort of sunk into this chronic problem. And we know it's problem. And I just want to, you've seen the data Dr. Halevi presented here. I just want to put 2 of the curves that you put up there, 2 of the slides. If you look at the top right-hand corner, we've had responses in all cell lines. And that's interesting. And again, I'm going to enter, every third word should be caution here. It needs to be in phase 1 studies. But this shows a really robust improvement in the platelet counts. Now, you need to understand this. You see, if you look at the bottom left-hand part of this top right-hand, the platelet counts are 42,000. Those are mean platelet counts. Several patients went down to even below 20,000, which are life-threatening situations. So this is really a patients who were deadly. They were probably getting platelets, transfusions every once or twice or more weeks here, and getting such a response here certainly is something that I'm going to use the word reassuring, because I'm going to be cautious throughout. If you go below that, and these are as objective data as you can get from phase 1, just looking at transfusion dependence, and to go down from a population in an intention to treat analysis of all at 21 patients, and knowing that when we started, we had 13 patients who were transfusion dependent, and then look at all the dropouts, some because they died early or dropped out, to only have 7 patients who are transfusion dependent, and that catches the eye. And so I'm going to say here, we have, again, I'm going to repeat the word where we don't have the control, it's cautious, but if you have a signal, and I'm going to say this is a personal opinion, it's not data, that it's a strong signal, and certainly something that would want me to look further into these data.
Thank you, Dr. Rowe. And with that, I would like to start to discuss the next steps. We are continuing to following these patients according to the protocol. We'd like to have a 1-year follow-up from last patient, for the last patient. In parallel to that, and based on the data that we're seeing in this study, the company is in a process to move forward, and our main goal is to initiate a phase 2 study during 2021 in the hematology field. We believe that we're seeing a good signal, and the ability of the cells to get the recovery of the hematopoietic niche and the free bloodlines. We truly believe that we might hold a product that can be a new standard of care for a variety of different hematological deficiencies, and we intend to push it forward, and you can expect to see a phase 2 in 2021 for the hematology field. And with that, I would like to hand it to Chen Franco, our CFO, to discuss financials.
Thank you, Yaki. So a short brief on the financial status of the company. So as of March 31st, Pluristem had $74 million cash on hand. On top of that, we expect to receive the first tranche of comprised of 20 million euros from the European Investment Bank by the end of first half of 2021. Together with the expected funds, we expect to have more than $98 million cash on hand that will allow us to continue the clinical development of our current products, current indication, but also will enable us to continue and further the development of this hematological program. Thank you very much.
Thank you. Ladies and gentlemen, at this time, we will begin the question and answer session. Analysts, by clicking the raise hand option on the bottom of the screen. Other participants, please click on the chat box and submit your question. For the question, please write your full name and company. Please stand by while we poll for your questions. First question is from Chris Howerton of Jefferies. Please.
Hi, good morning. Can you hear me?
Good morning, Chris.
Okay, great, wonderful. Well, thanks so much for hosting the call and taking the questions. Obviously, some very encouraging results here. So maybe just a few from me, and then I'll hop back in the queue. So first and foremost, I guess, what might we expect in terms of a registrational endpoint for this type of patient population? Is something like, the blood cell count's going to be sufficient, or would it be something in addition to that, something along the lines of perhaps transfusion independence or something like that? So that'd be one question. The second question that I had was related to the acute radiation syndrome program. I know that in the past there was some opportunities to pursue that with perhaps an accelerated path with the animal rules. So I just wanted to see maybe some updates on that side of the story. And then third question for me is related to the COVID trial, and just wondering kind of what the expectations we might have in terms of data disclosures, certainly with some locales being able to, do a better job than others and vaccinations. Just some updates on maybe some of your thinking there and expectations. And I really appreciate you taking the questions.
Thank you very much, Chris, for the questions. So regarding the first questions, what's going to be the primary endpoint for registration? So the best answer I can give that's definitely a discussion point with the FDA. We did see companies like Bluebird getting registration for a change in blood counts. That was through the EMA. And also we've seen cases that the blood products or reduction in transfusion is also maybe applicable. We are designing the protocol for the next one, and we're going to get to a meeting to confirm with the FDA. Both are very relevant, but I do want to get their opinion because before we are stating anything publicly. But the cases definitely have been there already. For the second question for the ARS, yes, we did generate over the years, both with the NIH and the DoD, quite a robust data. Post-exposure to radiation and pre-exposure to radiation. We are definitely going to do anything that makes sense in order to advance this program. I'm sure that showing clinical data is important, and we're going to give updates as we're going to have specifically for this product. But we're going to do anything that is possible that's necessary to advance this program. For the ARDS associated with COVID-19, we are running several multinational studies, and we're seeing the need in Europe, in US, and also in other territories, definitely. We would like to see, we had quite a robust data from compassionate use, but we would like to see it on a clinical data, the clinical data setting. And as I mentioned, with the timers that we presented, we would like to see that and to discuss with the regulators. Obviously, we have an open communication getting these studies approved with both EMA and FDA to get their opinion about potentially emergency use authorizations and things like that. And this is something that we intend to pursue specifically for COVID, and following that more broadly for ARDS in general. So this is on our map, and we're definitely going to do it once the clinical data will be available. We're seeing vaccination, but still we believe that this is a good time for therapeutics, treating the one that are either do not have access to vaccination or not responders to the vaccines.
Certainly. Okay. Well, thanks so much. And maybe I'm sorry, one other question did come to mind, if I may. With respect to the phase 1 study results, in terms of the dose selection that you have in this current study, it seems like we're starting to get more encouraging results with the higher dose. So I guess, is there any further dose exploration that might be incorporated into the clinical development plan moving forward?
So thank you for this question. This is Nitsan Halevi, the Chief Medical Officer. As you can see from the results of this study, of course, this was a dose escalation study, but a very small and preliminary one. And definitely, as we now plan our future clinical development, this is one of the things that we will explore in order to get as robust as possible design for our phase 2 that will allow us to better understand the dose response of our product.
Okay. Well, thank you very much. I appreciate it again.
The next question is from Mr. Matthew Kaplan of Langenberg Thalmann. Please go ahead and unmute yourself.
Hi, can you hear me?
Good morning, Matt.
Good morning. Good morning. Thanks, Yaki. Maybe first a question for Dr. Halevi and Dr. Rao. Can you give us a sense in terms of what you would be looking for in the 1-year follow-up from this phase 1 study, and what would get you excited given the initial 6-month results?
And this is Jacob Rowe. Thank you for this question. But obviously, the best that could happen is if this response is maintained at 1 year. That would be the best that we can have. But the course of these patients are such, there are multiple variables here, including one, often refractory disease that may relapse. And so there are conflicting variables here that go into place. But if I can only say, the most important thing that we're looking for is a sustained response in the presence of stable disease, that we don't have a relapse or other things that prevent this. But you want to see, obviously, the longer that this is not a transient response. I think that would be a crucial endpoint.
Okay, great. Thank you. And then kind of a follow-up on Chris's question, maybe more broadly, your thoughts on beyond dose in terms of the next study, the phase 2, what you're thinking of in terms of number of patients, duration of the study, and maybe some more details on your initial thoughts on the next study.
Thanks, Matt. So it might be a little bit premature to discuss that. We like to make sure that, first of all, we're going to have the FDA the chance to convey his opinion about the study before we're going to state it publicly. But we work on the numbers. We understand how it looks like. I think that it's quite easy to see how phase 2 studies in this indication looks like, but we do want to get the FDA opinion first in order to state that. But the good news that it's coming, and it's coming this year.
Okay, very good. And then last question, you mentioned in your prepared remarks that the muscle regeneration following the hip fracture study is about 85% enrolled. When do you think you'll complete enrollment in that study? And when should we expect some initial top-line results?
As I mentioned during the presentation, we expect to complete enrollment in the next quarter, so next couple of months. We're enrolling nicely. And the primary endpoint will be 6 months follow-up for efficacy. So this gives you more or less the time frame for readout. It's targeting like the first half of 2022.
Very good. Thanks for taking the questions.
Thank you.
The next question is from Mr. Arthur B. of H.C. Wainwright. Please go ahead and unmute yourself.
Hey everyone, thanks for taking my question. This is Arthur for RK. So I just want to follow up on the recommended dose question. So could we assume that we are not fixed on the 4 million dose level? And I noticed that for your ASH presentation, you mentioned the dose regimen is inject intramuscular in multiple locations. And could you give us more color on the dosing regimen in more details? Thank you.
Yeah, thank you. So, sorry for that. Again, as I mentioned previously, we did get some preliminary information about the efficacy of the study doses from the phase 1, but it is on a small number of patients and for a limited number of doses. So we may choose to continue exploring that. I don't think at this point it would be right to fixate on any specific dose. As for the dosing regimen, yes, the administration is through intramuscular injection in various locations in the body due to the limited ability to inject a high volume into one location. And the injections are provided at day 0 and day 7 again, so twice for each of the treated patients.
Oh, thanks for that. And just one quick follow-up. So as for now, how many patients still on the study?
Currently, there are 7 patients that we have data on after month 6, but several patients haven't had their month 9 and month 12 visits yet. The last patient out is anticipated in the fourth quarter of 2021, which is when we'll be able to do the complete analysis on all patients who reached the month 12 visit.
Thank you. Thank you for taking my question.
Thank you, Arthur.
And now we will continue with questions from the chat box. First question, can you please shed light on the potential of the addressable market?
This is Jacob Rowe again here. The potential here, I think, is actually probably more than we just comes to mind. Besides the graft failure that we've discussed, as I said, I think it's an increasing clinical problem as we're treating more older people, transplanting, a lot more people with comorbidities, and we're seeing it. But then there are a whole lot of other clinical scenarios, such as, for example, aplastic anemia. And then, probably intriguing, but if you take a leap forward, we use single hematopoietic targeted cytokine like GCSF or erythropoietins or thrombopoietin. Those are used regularly post-transplant. Now, if we had something that would be a pan-targetable, target several cytokines, that would be highly competitive. I think as a standard, if you're looking at potential out there, I certainly as a clinician, as a transplanter, would be very interested to know. What we usually do is we use GCSF because we're most worried about immediate infections, want the neutrophils up. We sort of pump the patients with platelets and red cells. If we had something that could increase all cell lines, I think, again, it's opinion, and this would be something that may be very competitive.
There are no further questions at this time. Before I ask Mr. Yanai to go ahead with his concluding statement, I would like to remind participants that a recording of this call will be archived and available on Pluristem's website, pluristem.com, beginning tomorrow. Mr. Yanai, would you like to make your concluding statement?
So thank you everyone again for attending this call. As we presented, Pluristem is ready for 21. The company is well funded with a series of clinical readouts coming this year. And with this data that we are presenting, we're definitely going to pursue the hematological field in also more intensively in 2021. So I would like to thank you all for attending here and being part of the process over the year. And I wish you all a great day. Thank you very much.
This concludes the Pluristem's call. Thank you for your participation. You may go ahead and disconnect.